Hello, I'm Ana Novoa and I'm going to present the dissertation called Carpobrotus edulis, a threat to the conservation of coastal dune ecosystems, with Dr. Luis González as supervisor. First of all, I will go over some background details. After that, I will ask the questions that we aim to answer with this thesis. Then, I will move on to explain each chapter, and subsequently, I will state our main conclusions. Finally, I will express my thanks to all the people who contribute to the development of this work. Well, considering that 71% of our planet is water, we can say that the Earth is a coastal planet. The coasts have always been the favorite place for humans to establish. The occupation of coastal dune ecosystems by infrastructures, cities, or industries have been the main causes of deterioration of these ecosystems. Moreover, the coastal dune habitats have to deal with the pressure of thousands of people every summer, and in order to prevent sand movements, we have planted pines or invasive species. As a result, only 45% of the coastal dune ecosystems worldwide remain in a natural way. The coastal dune ecosystems are particularly susceptible to the invasion by alien species. This is because the vegetation of these areas have to deal with a huge range of extreme environmental conditions, such as the low levels of nutrients on the soil, the temperature range between day and night, since during the day the sun can reach 60 degrees, salt spray, high winds, or, as we can see in the burial of this individual of Pancratium maritimum, sand movements that constantly produce open spaces susceptible to the invasion by alien species. The coastal dune ecosystems of the Iberian Peninsula have a huge range of different invasive species, as for example, Oxalis pescaprae, Arcoteca calendula, Stenotaphrun secundatum, Oenotera drummondi, Galenia pubescens, or Carpobrotus edulis, which is the focus of this work. Carpobrotus edulis is an invasive plant native from South Africa that invades coastal dune ecosystems in many parts of the world, such as, for example, New Zealand, Australia, Chile, California, or Southern Europe. When Carpobrotus edulis invades coastal dune ecosystems, it grows among and on native vegetation, creating a monospecific cloak and modifying certain quality soil parameters. For example, we can see in this picture sand from the same coastal dune ecosystem. The black sand is from an invaded area, and the white sand is from a native area. In the coastal dune ecosystems of the Iberian Peninsula, Carpobrotus edulis coexists with a hybrid, Carpobrotus athinaciformis. There is some controversy to distinguish them. For example, in the literature, we can find different features, as the color of the flowers. But, as we can see in this picture, we can find both colors in the same individual. Another feature is the cross-section of the leaves, but, as we can see, it is quite complicated to distinguish them by this feature. And finally, the color of the filaments of the stamens. This is the most reliable feature, so we have followed it in this work. Now, I will ask the questions that we aim to answer. In first place, what is the current situation on the Iberian Peninsula about Carpobrotus edulis? Secondly, what is the impact of Carpobrotus edulis on the soil, and how does this impact affect the establishment of the native species? In third place, does Carpobrotus edulis compete with native species? And going further, what other variables are important in the invasion of Carpobrotus edulis? And once we know all of that, if we decide to restore an area invaded by Carpobrotus edulis, is it enough to remove it? In order to answer the first question, what is the current situation on the Iberian Peninsula, we made a list of all the endangered species of the Iberian Peninsula. To do that, we used the red lists, red catalogs, and red books of Spain and Portugal. And once we had this list, we wanted to know the location of every individual of each endangered species, as well as the location of every individual of Carpobrotus edulis. To do that, we used ANTHOS, that is a program of biodiversity of Spain and Portugal. And then we completed this data with personal observations, interviews with local people, local herbaria, and so on. And once we have this database, as we know that the rabbits are able to disperse the seeds of Carpobrotus edulis up to 1 kilometer away, we consider that if we have an individual of an endangered species and within 1 kilometer away we have an individual of Carpobrotus edulis, this individual of endangered species is threatened by the presence of Carpobrotus edulis. So we put 1 if the individual of the endangered species is threatened by Carpobrotus edulis, and 2 if it's not. And we found that within 144 individuals of endangered species, in the Iberian Peninsula, 18 are not threatened by the presence of Carpobrotus edulis. 33 have less than 25% of their population threatened by the presence of Carpobrotus edulis. 38 between 25 and 50, also 38 between 50 and 75%, and 17 endangered species of the Iberian Peninsula have more than 75% of their population threatened by the presence of Carpobrotus edulis. Moreover, the areas with more number of endangered species on the Iberian Peninsula are Andalucía in Spain and Algarve in Portugal. But the areas with more endangered species threatened by the presence of Carpobrotus edulis are Cantabria in Spain and Baixo Alentejo in Portugal. So overall, we can say that Carpobrotus edulis is a real threat to the conservation of the endangered species of the coastal dune ecosystems of the Iberian Peninsula. So now, the next step is to answer the question, what is the impact of Carpobrotus edulis on the soil, and how does this impact affect the establishment of the native species? In order to answer that, first of all, we sample soils in the dunes of Punta de Ron in Ogrove, Pontevedra. That is the first place where Carpobrotus edulis was mentioned in Spain. We sample soils in an invaded area and in a close native area. And we analyzed the extracellular enzymes glucosidase, urease, and phosphatase, since they are responsible of the cycle of carbon, nitrogen, and phosphorus respectively. And we also performed an elemental soil analysis. If we see the results of the native areas, we find more or less the expected values for a primary dune, as for example, basic pH, low organic matter content, high salinity levels, low moisture, and low levels of phosphorus and nitrogen. This is because most of the nutrients necessary for the growth of the plants are included in the seawater, but the phosphorus and nitrogen levels are in such low amounts that they are a limiting factor for the growth of the dune plants. And as we found low levels of organic matter, phosphorus, and nitrogen, we also found low levels of the extracellular enzymes glucosidase, urease, and phosphatase. Now, if we compare those results with the results obtained in the invaded areas, we find significant differences in almost all the characters measured. For example, we found that Carpobrotus edulis decreases the pH. This is because this invasive plant takes large amounts of nutrients from the soil, decreasing the pH, and also it adds a lot of organic matter, a lot of litter to the soil. So the organic matter is higher, and this organic matter during the decomposition also decreases the pH. And moreover, the organic matter retains water on the soil, so the moisture are also higher in the invaded areas. The salinity levels are also higher in the invaded areas. This is because Carpobrotus edulis retains salt and marine debris on the leaves, and this salt then is transferred to the soil. And as I said before, Carpobrotus edulis adds large amounts of litter to the soil, so the phosphorus levels are higher in the invaded areas. But the nitrate levels are higher in the native areas. This is because the organic debris of the plants during the decomposition release ammonium. This ammonium is transformed in nitrite, mostly at pH between 6 and 9. In this nitrite is transformed in nitrate, mostly at pH 7. So, in the native areas, we have a pH around 9. So, the nitrite is not transformed in nitrate. But in the invaded areas, we have a pH around 7, so the nitrite is transformed in nitrate. And then, as I said before, Carpobrotus edulis takes large amounts of nutrients from the soil, so it assimilates the nitrate and the ammonium. So, we just found significant differences in the levels of nitrite. And then, the extracellular enzymes depend on the quality of the litter, and as we can see, we found higher levels of urease and phosphatase in the invaded areas. So, overall, we found more or less the expected values of the impact of Carpobrotus edulis on the soil, as we can see more or less the same in the literature. But we thought, is this the same in all the habitats? In order to test that, we compared the analysis of the dunes of Punta de Ron in Pontevedra with an analysis of different points all over the Iberian Peninsula with different climate, different distance to the sea, and so on. So, we compared the dunes of Punta de Ron, 10 meters from the sea, with dunes in Bilbao, 100 meters from the sea, dunes in Barcelona, also 100 meters from the sea, but in an urban area, in Alicante, 200 meters from the sea, semi-fixed dunes and with the influence of some pines around Carpobrotus edulis. In Huelva, also 200 meters from the sea, semi-fixed dunes, but with Carpobrotus growing among shrubs. And finally, in some dunes of Lisbon, it's fixed dunes, 400 meters from the sea, and Carpobrotus edulis growing among pines. So we have the results of the dunes, of the primary dunes of Pontevedra, Bilbao, and Barcelona, the semi-fixed dunes of Alicante and Huelva, and finally, the fixed dunes of Lisbon. And as we can see in gray, we found more or less the same impact of Carpobrotus edulis on the soil in the primary dunes. In the semi-fixed dunes, we didn't find a huge impact of Carpobrotus edulis on the soil, and in the dunes of Lisbon, fixed dunes with the influence of pines, where in the native areas we have a lower pH than in the other native areas, we found that Carpobrotus edulis in this case raises it. And the same with the nitrogen compounds. In this case, with the influence of the litter of the pines, Carpobrotus edulis decreases the nitrogen levels. And as I said before, the extracellular enzymes depend on the quality of the litter. So, as we can see, Carpobrotus edulis decreases the levels of glucosidase. This could be the reason why Carpobrotus edulis have low levels of descomposition rates. And also, the litter of Carpobrotus edulis increases the urease and phosphatase activity. But in Huelva and Lisbon, where we have the influence of litter of shrubs and pines, Carpobrotus edulis decreases the urease activity. So overall, we can say that Carpobrotus edulis have a different impact on the soil depending on the initial characteristics of the invaded ecosystems. Now that we know the impact of Carpobrotus edulis on the soil of the primary dunes, we want to test this impact on the establishment of the native species. To do that, we choose Malcolmia litorea and Scabiosa atropurpurea as target species. Malcolmia litorea is a species native from Spain, Portugal, Italy, and France. It's considered as an endangered species in some areas of Spain as well as in Italy, and is typical just from coastal dune ecosystems, while Scabiosa atropurpurea is from the Mediterranean and Macaronesian area. Is typical not just from coastal dune ecosystems, but also from ruderal habitats, and is considered as an invasive species in some areas of America. So, we collected seeds of these native species as well as seeds of Carpobrotus edulis. Also, we choose different levels of nutrient, salinity, pH, and moisture, depending on those levels that we found in the previous experiment. We made solutions with these levels, and we sown petri dishes with seeds of Malcolmia, Scabiosa, and Carpobrotus. So we have petri dishes, water with distilled water with pH 6 and 1 milliliter per day, distilled water enriched with nitrogen and phosphorus, distilled water enriched with different levels of salinity, with different pH, and also with 1 milliliter, 2 milliliter, and 3 milliliters of distilled water per day. And we recorded the germination rate, speed of germination, and radicle length. And we found that the germination rate of Carpobrotus edulis depend on the level of nutrients. This is because most of the dune species may benefit from growing in the vicinity of other plants because of the lower evaporation, lower isolation, and so on. So the selection process has favored the seeds to fill its neighbors. This could be the case of Carpobrotus edulis, but not of the native species Malcolmia litorea and Scabiosa atropurpurea. this native species, its germination depends on the levels of salinity and moisture. This is because Malcolmia littorea and Scabiosa purpurea used to germinate in autumn, the rainy season, when in addition to have more moisture on the soil, we also have low levels of salinity. But certain levels of salinity seems to improve the germination rate of Malcolmia littorea and Scabiosa purpurea. And finally, Carpobrotus edulis have higher levels of germination rate at pH of 8 and Malcolmia littorea at pH between 7 and 9. With the speed of germination, we found more or less the same, but we found that Malcolmia littorea germinates faster without nutrients on the soil. And the same with the radical length of Malcolmia and Scabiosa. This is because the coastal dune species can respond in a different way to the levels of nutrients. We can see again the effect of the salinity. And we found that Carpobrotus edulis and Scabiosa purpurea grow more with more moisture. But it is not the case of Malcolmia littorea, that is typical just from coastal dune ecosystems, with low levels of moisture on the soil. And finally, an interesting result is that the decrease in pH not only favors the growth of Carpobrotus edulis, but also of Malcolmia littorea. So, now that we know the impact of Carpobrotus edulis on the soil, and how does this impact affect the establishment of native species, we want to know if Carpobrotus edulis compete with native species. In order to test these competition relationships, we choose again Malcolmia littorea and Scabiosa purpurea as target species. And we establish competition experiments between adult individuals of the native species and ramets of Carpobrotus edulis. We establish a replacement series experiment, mixing one individual of Carpobrotus edulis with 3 individuals of native species, 2 ramets with 2 individuals, and 3 ramets with 1 individual, in both invaded and native soil. And in order to take into account the intraspecific competition, we also establish pure cultures of 1, 2, 3, and 4 individuals of native species and 1, 2, 3, and 4 ramets of Carpobrotus edulis. But we didn't find intraspecific competition, and we didn't find any effect of the soil on the monocultures, on the pure cultures. But in the mixtures, here where we have the results of Malcolmia littorea, for Scabiosa it was the same, we have number of live adult plants every 2 weeks. We can see that Carpobrotus edulis was detrimental for the native species, and this effect was even faster in the invaded areas. But in both invaded and uninvaded areas, Carpobrotus edulis killed all the native individuals. So, once we know the effect of Carpobrotus edulis on the adult plants of the native species, we want to test the effect of the presence of Carpobrotus edulis on the establishment of Malcolmia and Scabiosa. To do that, we collected seeds of Malcolmia and Scabiosa, and we established a competition experiment between those seeds and the ramets of Carpobrotus edulis, following the same scheme as before, but this time with number of seeds of native species, 10, 15, 20, and 25 seeds instead of 1, 2, 3, and 4 individuals. And we collected the data of germination rate, speed of germination, root growth, and survival rate. This time, in the monocultures, we found, surprisingly, that the germination rate and early growth of Malcolmia littorea was better in the invaded soil than in the uninvaded soil. But in Scabiosa purpurea, we didn't find significant differences. This could be because, as I said before, Scabiosa purpurea is typical not only from coastal dune ecosystems, but also for ruderal habitats, so probably it is more plastic than Malcolmia littorea. In the mixtures, we found that the presence of Carpobrotus edulis, here where we have 10 seeds with 3 ramets, 15 with 2 ramets, and 20 with 1 ramet of Carpobrotus edulis in invaded and native soil, we can see that the presence of Carpobrotus edulis was detrimental for the germination of Malcolmia in the uninvaded areas. But the most interesting result was that the presence of Carpobrotus was detrimental for the survival of Malcolmia in the invaded soils. But it killed all the native seedlings in the uninvaded soils. And once again, we didn't find significant differences in Scabiosa purpurea. So, now that we know the effect of the presence of the ramets of Carpobrotus edulis on the native species in all the stages of development, we want to know the competition relationships established between seeds of the native species and seeds of Carpobrotus. So we collected the seeds of both species. And we established a competition experiment mixing 10 seeds of a native species with 10 seeds of Carpobrotus, 30 with 10, and 10 with 30. And we also established monocultures of 10 seeds and 30 seeds of each species in both invaded and native soil. But we also wanted to know if the seeds are able to feel each other. To do that, we sown Carpobrotus edulis 5 days before the native species, at the same time, and 5 days later. And we recorded the germination rate, the speed of germination, and radical growth. This time, as we are using sand in the petri dishes, we had to make holes in the bottom of the petri dishes in order to prevent the formation of salt crust. And here, where we have the results of Carpobrotus edulis in the presence of Malcolmia, Carpobrotus in the presence of Scabiosa, Malcolmia, and Scabiosa, in both treatments where Carpobrotus edulis was sown 5 days before, at the same time 5 days later, and in the monocultures, we can find that Carpobrotus edulis germinate better and faster where it was sown 5 days before Malcolmia or in the monocultures. And with Malcolmia it was the same. Malcolmia germinate better when Carpobrotus edulis was sown at the same time 5 days later or in the monocultures. So we can say that Carpobrotus edulis and Malcolmia litorea can feel each other. In the early growth, we didn't find significant differences. So now that we know the impact on the soil, how it affects the establishment of the native species, the competitive relationships, we want to go farther. As we saw before, we know that Carpobrotus edulis have some effects on the soil characteristics, and these effects have an effect also on the establishment of the native species. But we want to know if those characteristics have a synergistic effect on the establishment of the native species. Moreover, as I said before, the rabbits are able to disperse the seeds of Carpobrotus edulis. But we want to know if those rabbits have an effect on the establishment of the invasive species. And also, as I said before, Carpobrotus edulis adds large amounts of litter to the soil. We want to know if this litter has an effect on the establishment of the native species. So, in order to look for those synergistic relationships between the characteristics of the soil on the establishment of the native species, we made solutions mixing the values of pH and salinity that we saw before that affect the establishment of native species. So we have solutions with pH of 7.5 and low levels of salinity, pH of 7.5 and high levels of salinity, and the same with pH 8.5. And once we have those solutions, we sown petri dishes with 10 seeds of Malcolmia or 10 seeds of Carpobrotus, and we water them with 1 milliliter or 2 milliliter of each solution in order to take into account also the effect of the moisture. And we recorded the germination rate, speed of germination, and radical growth. But we didn't find significant differences about synergistic effects. We didn't find significant differences with the levels of pH or the levels of salinity, but we found significant differences with the level of moisture, as we can see here in the results of Malcolmia litorea, where the treatments with high moisture present higher values than the treatments with a low moisture in both germination rate and root growth, and the same with the root growth of Carpobrotus edulis. So, we can say that the moisture is the most important characteristics of the soil affecting the establishment of Malcolmia litorea and Carpobrotus edulis. The next step is to know if the rabbits have some effect on the establishment of Carpobrotus edulis. To know that, we feed rabbits with seeds of Carpobrotus edulis, we collected the excrements, and we collected the seeds on the excrements. And we found that the scarified treatments have higher values than the unscarified ones. So the rabbits not only disperse the seeds, but also improve the establishment of Carpobrotus edulis. And finally, the litter. To test that, we collected rainwater in native dunes, dunes invaded by Carpobrotus edulis, and dunes where Carpobrotus edulis was removed 1.5 years before and 3 years before. To collect this rainwater, we removed the litter, removed the sand, we put a plastic tray on it with a nylon net, and we put on the plastic tray the sand and the litter, and we collected the rainwater. We didn't find significant differences in the levels of pH or conductivity between the 4 rainwater solutions. And once we have those solutions, we sown petri dishes with seeds of Malcolmia and seeds of Carpobrotus. And we found no significant differences on Carpobrotus edulis, but we found that the rainwater solutions collected in those areas where Carpobrotus edulis was removed were more detrimental for the germination rate and speed of germination of Malcolmia litorea than the other rainwater solutions. This could be because the accumulation of different compounds during the descomposition of the litter. But in the root growth, we found the opposite effect. This could be because the accumulation of different nutrients on the soil during the decomposition process. So now we know the impact of Carpobrotus edulis on the soil. How does this impact affect the establishment of the native species? We know the competition relationships between the native species and the invasive species Carpobrotus edulis, and also we know the effect of the rabbits on the establishment of Carpobrotus edulis and the effect of the litter of Carpobrotus edulis. So now we want to restore an area invaded by Carpobrotus edulis. We know that in a lot of areas where Carpobrotus edulis was invading, removal projects have been carried out in order to restore the invaded areas, but they have not achieved the ultimate goal of dune restoration. In those areas we can find Carpobrotus edulis reinvading or a plant community completely different to those species that we find in the coastal dune ecosystems. To know what's happening here, following Conser and Connor design, we established 9 plots in an area invaded by Carpobrotus edulis and 9 plots in a native area. Each plot was subdivided into 3 subplots. The first one was sown with Malcolmia litorea seeds. The second one was transplanted with Malcolmia litorea seedlings, and the third one was unplanted. So with the first and the second one, we could compare the growth of Malcolmia litorea growing from seeds and from seedlings. And we didn't find significant differences. So in our restoration, we could sow instead of transplant, since it is cheaper and easier. Moreover, comparing the growth of Malcolmia litorea and the germination and survival in invaded and uninvaded areas, we found that the invaded areas are detrimental for the germination and mortality of Malcolmia litorea. But we didn't find significant differences on the growth. So in a restoration, we must overseed, since once the native species are established, the growth is equal in native and invaded areas. And then the third subplot. We want to know what's happened in these areas if we don't sow or if we don't transplant. So we analyzed the biodiversity. And we found that the Shannon-Simpson and Margalef indexes were significantly higher in the native areas than in the invaded ones. And moreover, we found a higher number of species and individuals of dune species in the native areas than in the invaded ones, and a higher number of individuals of ruderal species in the invaded areas. To know what's happening here, we established a competition experiment between the ruderal species and native species that we found in the previous field experiment, the most common ones. So we choose Scolimus hispanicus and Dactylis glomerata as ruderal species, and Malcolmia litorea and Caquile maritima as native species, and we established this competition experiment in both native areas, native soils and invaded soils, and in invaded soils plus litter of Carpobrotus edulis, to take into account also the effect of the litter. So, for example, in the case of Malcolmia litorea, we have pure cultures of 14 seeds of Malcolmia and mixtures of 7 seeds of Malcolmia with 7 seeds of Scolymus and 7 seeds of Malcolmia with 7 seeds of Dactylis in both native and invaded soils, as well as in invaded soils plus litter of Carpobrotus. And in this table, we have the results of Malcolmia. With the other native species, it was the same, where we have the results of the native and invaded areas in pure cultures, mixtures with Scolymus, mixtures with Dactylis, and for the germination and early growth. And as we can see, the competition relationships established between ruderal and native species were more detrimental for the native species on the invaded areas than in the native ones. But this effect was even more dramatic for the native species in the invaded soils plus litter of Carpobrotus edulis. Finally, we also wanted to know if the soil are able to follow a natural restoration process after the removal of the invasive plant. To do that, we analyzed the soils of native areas, invaded areas, and of those areas where Carpobrotus edulis was removed 1.5 years before. And we analyzed, once again, the extracellular enzymes glucosidase, urease, and phosphatase, responsible to the cycle of carbon, nitrogen, and phosphorus, and we also performed an elemental soil analysis. And we found that the microbial community are able to follow a natural restoration process after the removal of the invasive plant, since the values in the native and restored areas were lower than the values in the invaded ones, and more or less the same with the characteristics of the soils. But the pH and nitrogen levels didn't follow this natural restoration process. This persistence of pH and nitrogen levels could be the reason why the ruderal species are able to compete with native species after the removal of the invasive plant. So, the conclusions of this thesis are, related to the questions that I asked at the beginning of this presentation, are, in first place, that Carpobrotus edulis is a real threat to the conservation of the endangered species of the Iberian Peninsula. Then, Carpobrotus edulis has an impact on the soil, and this impact affects the establishment of the native species. And moreover, Carpobrotus compete with the native species in all the stages of development. The rabbits not only disperse the seeds of Carpobrotus edulis, but also improve its establishment, and the litter has also an effect on the establishment of the native species. So, if we want to restore an area invaded by Carpobrotus edulis, we have to remove not only the invasive plant, but also its litter, to overseed with native species, and to remove those individuals of Carpobrotus edulis and ruderal species that will be able to establish in those areas. Finally, I want to express my thanks to all the people who contribute to the development of this work, so thanks to you all. And also to say that this work resulted in different communications to several national and international congresses, as well as in 6 papers. Thank you so much for your attention.